Okay. Hey, so we'll start. Uh, this is live session number 7, and we are discussing week 7 lectures. Note that we'll have a final revision class, which is scheduled to be held on 20th March, 2024, which is Wednesday, from 6 to 8 p.m. So we'll discuss all the material for the course, and if you guys have any doubts, you can uh, ask right before the exam. Okay. Here are some important links and some other important links. So this link, this is the link to download the codes for the course. So you can go there and download all the codes and the data that is used throughout the course in different lectures. Uh, so today we'll discuss about week 7, which is clustering and classification. So I discussed about k-means clustering algorithm. So k-means clustering is basically an unsupervised statistical learning method where we just have some data points, but we don't have any labels for the data points. So what does k-means do? It basically it's a iterative process where you repeat a certain number of steps, uh, certain steps a certain number of times, and then so basically in one step you assign points to cluster, and in the second step you find the center of the cluster and find distance to different points. So we'll go through the algorithm first. So let's say we have data like shown on this on the top right corner. Let's say we have like these 4 points, 3 points, and 4 points here, and we want to cluster them. So what we'll do, so let's say we want to make 3 clusters. So what we'll do, first you specify the number of clusters, which is k. So in this case, we want k to be equal to 3. Okay, because we can visually see that the data has might be in 3 different clusters. Then what you need to do, you need to first randomly assign some centroids. So since k is 3, we'll randomly assign 3 centroids. So let's say one is here, one is here, and one is here. Next, what will happen? So we need to calculate the distance of every data point from each of these centroids. So distance between these two, these two, these two, these two. For all these points, we have to do that. And after that, what we do, for each data point, we figure out which is the closest cluster center, which is the closest centroid to them, and then we assign it to that cluster. So basically for this point, visually we can see this is the closest point. So this would go to this cluster, this point would go to this cluster, this would go to this cluster, and this would go to this cluster, right? Similarly, this point will come here, this point will come here, this point might go here initially. Then, so basically we'll draw 3 clusters. So let's say this is all the points that are assigned to the first cluster. Okay, then these are all the points assigned to this second cluster, and this is the point that is assigned to the third cluster. So we have 3 clusters. So next what we do, so in the next step what we'll do, so next step we basically have the same data again, which is 4 points here, 4 points here, and 3 points here. Right. So in the next point, what we do, these all data points that belong to this cluster, first cluster, we'll find a centroid based on the position of these data points. Basically, we'll find a centroid that is basically the average position of this cluster. So let's say we get something that is here. So that will be a new centroid for this cluster. Similarly, we do this for this. So it comes on top of this data point. Then for this one, if we do this, it should come somewhere here. Then what we do, we again like calculate the distance from the clusters, assign each data point to a given cluster, then again calculate the centroid another time and go on. And this step basically repeats. So this is an iterative process. So how long till do you have to repeat this for? So you repeat this till your centroids are fixed. So for example, here the centroid, initially the centroid might have been here, but it moved to here. And initially this one might have been here, but it moved to here. And this one was probably here, and it moved to here. So when your centroids, like after you do enough number of iterations, your centroids will eventually stop moving. So when you do that, you basically you have proper clustering of your data in this algorithm. So what you do, you stop there. Otherwise, you have some set number of repetitions that you want to do. So let's say I want to just repeat it 500 times or something. I think that's enough. So you do that. This is the basis of k-means clustering algorithm. Now, how do we evaluate the results of a clustering? So as mentioned before, it's an unsupervised learning method. So we don't have any labels or anything. So how do we judge if whatever cluster we are getting is it is what it represents what clusters are there in the real data? So for that, what we do is we calculate something called within cluster sum of squares. Okay. So basically, let's say we have some data like this. Okay. Then we have these three clusters and we have their centers here. So let's say this center is here, this center is here, and this center is here. So within cluster sum of squares is basically for each of these centroids or each of these centers of clusters that we have, we calculate the distance from the points present in the cluster. So this distance, this distance, this distance, you calculate the distance, square it, and add it. You do the same for here. You calculate this distance, this distance, this distance, all four, square it, add it. Then again, these four distance, calculate, square it, add it. So basically what we are doing is we are quantifying how much spread out each of our cluster is. So this within cluster sum of squares will be greater. For example, if you have a, let's say you have two types of clusters. You have one cluster here in which all the data points are very close by. And then you have another cluster here where all the data, like some of the data points are slightly farther away. So in this case, you will get a higher value of uh, within cluster sum of squares. And here you'll get a lower value of within cluster sum of squares. So basically what we want to do is we want to minimize the within cluster sum of squares so that we get clusters that are like tightly bound together or more likely to be homogeneous. Okay. Uh, so this is basically the formula. It's written mathematically how to do it. Uh, to choose like another thing that we need to answer is how do we choose like uh, okay, before that, so basically, where do we use this within cluster sum of squares? We use this when we are comparing different iterations of the same model. So for example, so let's say in this k-means algorithm, the first thing was specify a number of clusters and then randomly assign centroids to each for the clusters. So in this iteration, when we ran the model, we initially assigned the centroids here, but we could have assigned the centroids somewhere else also, let's say here, here, and here. That would also have been fine, and that would probably have led to different results or different clusters. So to get like, so because there is some variability in this algorithm because we are randomly assigning cluster centroids in the beginning, which might lead to different results. That's why we need this within cluster sum of squares to compare all the different models. So what you do basically is you basically run 10 models for one k value and then choose the model that has the lowest within cluster sum of squares. Okay. And then another place where you can use this is basically when you are choosing the optimum number of clusters. So again, since it's an unsupervised learning method, we don't sometimes we don't know how many clusters might be there in our data. So basically, this data is very sparse, so you can easily say that how many clusters are there. But what if the data was something like this? So it's slightly more complicated and we don't know exactly how many clusters are here. So what we do is basically we try out different values of K. We try out different numbers of clusters and then we calculate for each of the runs of the model for each of the value of K. Let's say 1, 2, 3, 4, 5, 6. We test it. We get what is the value of this within cluster sum of squares. We plot it. And this plot is basically called a elbow plot. Okay. So what it basically shows is how does the within cluster sum of squares decrease as we increase the number of clusters. So of course, when you have one cluster, it's very high because that is equal to the variance of the data. When you have two clusters, it's slightly less because the variance is now distributed within two clusters. So this thing keeps on decreasing. And what we need to do is basically, so let's say you connect all these together to draw a line. Then at some point, this line will start to asymptote. So basically, the line curve flattens out. And so that is the place. So basically, 4 is where the curve is flattening here. Okay, after this, the slope is decreasing. It's the inflection point. So basically, 4 is the number of models we should choose. Because when we are choosing, 4 is the number of clusters that we should choose. Because when we are choosing 5 clusters, we are just getting slightly better results, which might not be worth using a higher number of clusters. Okay, so it's not adding that much to our model. So you want to stop here. And this plot, because of the shape of this, so it generally looks something like this. So it's probably because of the shape of it, it's called a elbow plot. Does anyone have any doubts regarding this? Okay. So we'll move on and see how to do this in R. So how to do k-means clustering. So first we'll read some data. Sorry. Which is basically P1, P2, we are given two variables. One will plot on the x-axis, one will plot on the y-axis to plot the points. So first we'll make a plot and see how the data is. So plot, you give it the data that you want to plot. PCH is basically the symbol, which is empty circle. CEX is basically size, increase the size. So if we zoom on it and see, you see that our data is distributed somewhat like this. So probably it looks like there might be 1, 2, 3, 4 clusters or something, 4 or 5 clusters. Right. So what we do first now, first we perform k-means clustering. So first we'll start with two clusters. So k-means clustering can be performed using the k-means function in R. So you provide as the first argument the data. Then 2 is basically your number of clusters you want. N start equal to 50 basically. So in k-means, what we do, we initially randomize the centroids, no? So I told you what you need to do is you need to run the model a few times and then choose the best model. So N start basically does that. So it will run 50 models with initially random values, then it will choose the best model for you. So ideally, when you are using it, anything above any value of 20 or greater is okay. But in this case, we have used 50. So we did this, came. So now we see what is in there. So it has different things. So cluster is basically an array. We'll take a look at it. Uh, Vector. So cluster is basically the vector describing all these points that we have in the data. Which cluster do they go to? So for example, the first point belongs to cluster 2. The second point belongs to cluster 2. Third is also cluster 2. Whereas the last data point, 89, which is, it belongs to cluster 1, and so on. Okay. So now what we'll do, we'll basically plot this data again, but color it according to the cluster. How can we do that? So we use the plot function You provided the data. PCH is basically setting the type of symbol. CX is basically increasing the size. And call here is basically what color we want it to be. So we want the color to be KM2 dollar cluster. So whatever was in there. So it has all the cluster like numbers for all the data points. So basically it will pick two colors, one for two, one for one, and it will color it accordingly. So if you run this code, we see that our plot has been colored. Right. So basically we have two clusters. One of them is on the bottom left corner. The other one is somewhere near the right hand side of the image, right and up. Okay. Then what we want to do, we want to add the centers of these clusters. We want to see where the centers are. So in KM2, we have another variable called centers, which basically contains the coordinates of the centers. So what we do, we use the function points to add points to our plot. We provide the coordinates using KM2 dollar center. Color is 3. So you want it to be some a third color. Then PCH is basically the type of symbol and CX is the size. We run this, we see that we get like two centers of our cluster, which are basically one is here and one is here, almost at the middle of all the data. Okay. Then we want to do it again with 3 clusters and see what happens. So what we'll do, we'll use again the k-means function. We'll now provide it the value 3 because we want 3 clusters. We run this, then we plot it, and again coloring using the k-means $3 cluster variable so that we get the color of the clusters. You see that our data is like this. So we have 3 clusters. This is one. These two have been divided into 2 clusters. This one, the whole one was a single cluster before. Okay. Now we want to do it with 4. So again, we replace it with 4. Then we color it according to the cluster and plot it. So now we have 4 clusters on 4 parts. This is cluster 1, cluster 2, cluster 3, cluster 4. Okay. So as I mentioned before, how do we know what is the optimal number of clusters? Because we just have this data. We don't know exactly what the clusters are. So what we'll do now is we'll make the elbow plot that we were talking about. Okay. So first, to make the elbow plot, we need to carry out the k-means clustering for different values of k. So we'll go till 7 here. So we have done till k4, k is equal to 4. So we'll do 5, 6, 7, 5, 6, 7. Then, as I mentioned before, sorry, as I didn't mention before. So basically, this thing, so this KM2 has different variables. So this total dot within SS is basically the within cluster sum of squares, the thing that we want to minimize. Okay. Between SS is basically the uh, sum of squares between the different centers or between the different clusters. What is the separation between the different clusters? How far are they? How far apart are they? Okay. So basically what we'll do from each of the model KM2, KM3, KM4 till KM7, we'll extract this tot dot within SS because these are within cluster sum of square. We extract it into different variables W2, W3, W4 till W7. Then we'll make a bar plot. The values will be this W2 to W7. Names will be sequence 2 to 7. So basically we want the names on the for every bar to be 2, 3, 4, 5, 6, 7. Okay. On the x-axis, we want the label to be k. On the y-axis, we want it to be within clusters sum of squares. And y limit we have put from 0 to 4. If we run this, we see that this is the plot that we get. You see that as we increase the number of clusters, initially the within cluster sum of square decreases rapidly, but then after some time it slows down. So basically, looking at this plot, if we want to stop somewhere, we can a good place to stop would be 5 or 6, because after that the decrease in within cluster sum of squares is not that significant. So this is our elbow plot from where we choose what is the k that you should take for your data. Right. So looking at this, we see probably we should take 5 or 6 as the answer. Right. Then what we do, we also want to plot the between cluster sum of squares. So as I was mentioning before, this within cluster sum of squares is basically variance in a cluster, inside a cluster. This between cluster sum of squares is basically a measure of how far these clusters are. What is the distance between, like, let's say these 3 clusters? How far apart or how well separated are they? So we want to maximize this value because we want to maximize the separation while also minimizing the clustering within. Uh, so we want to maximize this separation within the centroids, but we also want to minimize the spread around the centroid, right? So the first plot that we saw here, we are trying to minimize the spread around the centroid. Now we want to see what the other plot looks like. So again, we'll get the between sum of squares from the km variables, and we'll plot a bar plot again. And this is basically the between cluster sum of squares, which we ideally want to maximize. So here you can see as you go from 2, 3, 4, the value increases quite rapidly. But however, after 5, the value does not increase that much. So according to this plot, we should go with number 5. According to the previous one, we should go with number 5 or 6. So in the end, we decide we should go with number 5. Okay, because that seems like it balances out both of them. Now what we do, we plot the data for the selected thing. So basically, we plot the data and color it according to the fifth claimants. So according to a k equal to 5 model, and then use the cluster information to color the data. So basically what we get are 5 clusters, one here, one here, one here, one here, and one here. Now, let's say you want to extract data for a particular cluster. So let's say this P1 and P2 are different genes, like basically gene expressions. And basically all the points that you have basically tells you how expressed this gene is in a given cell versus how expressed this gene is in a given cell, P1 versus P2. So all these data points are, let's say, different cells or something. And after we did a clustering, we now know that these cells, these red ones or these black ones, probably behave similarly. So that's why they are closer together in space, right? So what we want to do, we want to take all these points out from the data, and then we want to study it further, let's say. So how do you do that? So basically, you first select the indices for the data points in a cluster. So which KM, KM5 cluster equal to 1 will basically give you where all. So I'll just show it, right? C1. Okay. So KM5 clusters. So this is basically a 89 element vector. Okay. Which contains all the data for cluster. When we are doing this, what we are doing, we are extracting indices for data that belong to cluster 1. So which KM5 cluster equal to 1. So we get all the indices of the points that are there in cluster 1. Then what we do, we store it into, we create another variable cluster.1 where we take these points from the data. So we have the data. So we want these rows. So these are row indices. So we'll put it here, and we want all the columns. So we'll put a blank here. And when we run this, we get cluster.1, which is basically 14 observations, all the points that fall into cluster 1. Okay. Does anyone have any doubts regarding this? Is this clear? Okay. So we'll move next to hierarchical clustering. So hierarchical clustering is another clustering algorithm that we learn. So k-means clustering is basically flat uh, clustering technique. However, hierarchical clustering is a hierarchical clustering technique. So it does not just give us different clusters, it also gives us the relationship between different clusters in terms of their dissimilarity or distance to each other. Okay, which is shown by a dendrogram. Uh, One special thing is that in this case of hierarchical clustering, we don't need to mention any number of clusters. So for k-means, we had to specify how many clusters we want in our data. However, in hierarchical clustering, we don't have to do anything of that sort. Okay. So it's also agglomerative clustering technique. So what it means is basically, initially it will consider all the points as independent clusters. So now what we have here is A, B, C, D, we have 4 independent clusters. Then what you do, so when you're carrying out hierarchical clustering, what you do next is you measure the distance of each cluster from each other. So basically in this case, A, B, C, D are the different clusters. So A to B distance I want, A to C, A to D, B to C, B to D, C to D, all the distances we calculate. Then we find the two closest pair of clusters. So that will be A and B. Guessing on the visually just taking a guess, it should be A and B. Okay. So then we combine A and B to form a new cluster. Let's say we name it AB. Okay. Then what you do, you build a dendrogram. So a dendrogram is basically, let's say you have, huh. So since we are merging A and B, what we do, we put A and B and then we connect them like this. In a dendrogram, all these things, all these data points that we have, the final data points, will be at the leaf nodes. So they'll be at the end of it, and our tree will build up from here. And basically, the height of everything we scale based on the distance. So for example, this will be distance between A and B divided by 2 because it goes like this, right? Then we did this, we build a dendrogram. So the next thing is we have to repeat the second step again. So these three steps we need to keep repeating. So then what we do, we then find the closest pair of clusters. So now we have A, B, C, and D. We measure the distance between A, B, and C clusters and A, B, and D, and C and D. And let's say we figure out that C is closer. So now we have another cluster, which is this, which is basically ABC cluster. Okay. So now what we'll do, we'll add a third point here. We'll again build a dendrogram. We'll add C here. And basically, we connect it like this, where this distance will be distance between AB and C divided by 2. Okay. Then we again repeat it. So now we just have one point D to cluster. We just cluster it. The whole thing becomes one cluster now. And we plot it like this. Basically, this is T, and this distance will be distance between ABC cluster and D. divided by 2. Right. So this is basically our hierarchical dendrogram. So this dendrogram shows us what the relationship is between different clusters, how far apart they are, and which one is connected to which one. Okay. So while we are doing this, one of the steps that we need to do is basically we need to sometimes figure out what is the distance between uh, a cluster and a point, or between two clusters. So for example, here in the second step, when we figured out the distance, when we had to check the distance between A, B, and C, what we are doing is we are checking the distance between this cluster AB, both the points AB against C. So how do we need some way to measure that distance? So this distance between two clusters is also called the linkage. And there are basically different ways to different measures of linkage. So the first one is called centroid linkage. So it's quite straightforward. So let's say you have some data like this. You have points here that belong to one cluster and points here that belong to another cluster, right? Then what you do in centroid linkage, you figure out where the centroid of this cluster is, which is basically the average of all the positions of all the data points. Then you figure out where the centroid of this is, and you basically take this distance, the distance between this centroid and this centroid. So that is your distance between these two clusters. Okay. So that is centroid linkage. For the rest three linkages, what we do, we don't take the centroid. So let's say we again have some similar data. Okay. So now what we want to do, we want to calculate all the, so this is basically our cluster one, this is basically our cluster two. We want to calculate all the pairwise distances between elements. So basically distance between these two, these two, these two, these two, then again these two, these two, these two. All the distances for all of these uh, points in each of the cluster, we calculate all the distances. Okay, so we did that. Then single linkage is basically, so you'll have like a list of distances, or you'll have different distances. No. So let's say we have distance 1, 4, distance, sorry, 1, 5. Let's say this is 1, 2, 3, 4. This 5, 6, 7, and 8. Distance, we'll have distance 1, 5, 1, 6, 1, 7, 1, 8, 2, 5, and so on. So all these distances we'll have. So when you take the minimum of those distances, so let's say distance between 3 and 5. So D3, 5 looks like it will be the minimum distance. So this distance basically, I'll draw it again. Let it be. So this is 1, 2, 3, 4, there's 5, 6, 7, 8. So this distance between 3 and 5, this is the minimum distance between these two clusters. So that distance, if you take that distance as the distance between the clusters, then we term it as single linkage. Okay. Now, to find out what is the complete linkage, you take what is the maximum of the distance. So in this case, the maximum distance might be between 1 and 7. So distance 1 and 7, that will be the maximum distance. So this will be our complete linkage. Okay. And average linkage, as the name suggests, is basically the average of all these distances that you have across all the clusters. Okay. So the outcome of clustering, when you're clustering anything, will always depend not only on the measure that you are using to find out the distance between different data points. So for example, when you are calculating distance between these 2 points or these 2 points or these 2 points initially, when you are making the cluster, so these distances, or when you're using the distance thing in k-means. So this distance can be Euclidean, Mahalanobis, or some other kind of distance also. So depending on the type of distance you choose, your clusters might your clusters that you get we might be different for these different kinds of distances. And also based on all the linkage measures that we discussed here, the centroid linkage, single linkage, complete linkage, and average linkage, based on what you choose for your data, your clustering outcome will also depend on that. So you might get different cluster of points if you take different kinds of distance or linkage measure. Okay. Is this clear for everyone? Does anyone have any doubts? Okay. So we'll move on to R, where we also sir showed how to basically carry out hierarchical clustering in R. So basically, we read some data. Let's see what the data is. So basically, this is data of cells taken from different cancer cells. So uh, readings taken from different cancer cells. So basically, this is from CNS cancer, this is from breast cancer, this is from renal cancer, and so on, ovarian, prostate, melanoma, leukemia. And these columns are basically X1, X2 till X49 are basically different cells, different genes, their gene expression. So we have gene expression profiles for different cells we have taken from different cancer cell cultures. Now, what we want to do, we want to cluster them. Okay, we want to cluster them and figure out which of these cells might behave are behaving similarly, right? So first, we need to prepare the data. So when we are doing clustering, we just need this part of the data. We don't need the labels. So we'll separate out the labels to another variable. So basically what we are doing here, g dot lab is basically labels. So we want all the rows. So we left this empty and we just want the first column which contains the labels. So you do this and you get g dot labels, which is basically a vector containing character vector containing all the labels, right? Then we get the gene expression data. So for that, what we do, we select from G all the rows because we are giving keeping it empty here. And here, by writing -1, what we are saying are we are saying that leave the first column and get everything else. So everything minus the first column. Okay. Now we run this and we see G dot data is basically now a data matrix. It does not have the labels. Right. So before we carry out any hierarchical clustering, first thing we need to do is we need to scale our data. So what does that mean? So basically we make our data in each column. So for each of these genes, we need to, because we are comparing across these genes, we need to standardize the values that are there somehow. Normalize or standardize the values. So what we'll do basically for each of the gene expression, for each of the genes independently, we'll make sure that the mean is 0 for all the data points. The mean is 0 and the variance is 1 so that it's comparable across different genes. So how we do that? We basically subtract the mean from each of the data points and we divide it by the variance. So that's called scaling or normalizing the data. In R, we can do it using the function scale. So you just pass the data to the function g dot data inside scale and you store it at g dot sc. Okay. Next, what we do, we have to calculate the pairwise distance between all the observations. Before we do hierarchical clustering, we need to calculate all the pairwise distance. So we can do that using the dist function. Dist, we provided the scale data that we have. Okay. And we want to use Euclidean distance. So we provide method equal to Euclidean and we carry this out. So we get something called g dot test, which is basically a matrix kind of, which tells us what the distance is between different different samples, basically. Sorry, different genes, basically, different columns. Okay, it should be total of 63. 63 columns. So you should have that many columns. 64. No, 64. Okay, so it's basically for the samples, distance between each samples. Sorry, yeah, that was the whole point. Yeah. So notice that it's showing up as a square matrix kind of thing, but when it's stored in R, this variable, it's basically a sequence, number sequence, sorry, which has 1, 2, which has 20, 16 values, basically. Okay. So this is because we are basically taking pairwise distance between each observation. So we are making pairs out of observations. So when you do that, basically what you get is these many values. So the formula is n into n minus 1 by 2, right? Because so when we are checking the pairwise distance, let's say we have 3 points here and 3 points here. We're checking the pair. So basically we have 3 points in total. We want to check the pairwise distance. So we don't want the, so this is 1, 2, 3. This is again 1, 2, 3. We don't want the distance from 1 to 1. It will be 0, of course. So we want this distance, this distance. For 2, we got one distance. So we just want this distance. And for 3, we don't need because 1 to 3 and 2 to 3 is already accounted for. So in case of 3 points, you have 3 into 2 by 2. So basically 3 distances. So this is like n into n minus 1 by 2 is like a generalized formula for how many of these you will get, basically. So when you do that for a data set, which contains 64 genes, so 64 into 63 by 2 comes out to be 2016. Okay. Yeah. So after we have this distance, pairwise distance of all the points, we use it in the hclust function in R. So hclust, we provided the distance vector containing all the distance, and we tell it that use average linkage. Okay. So method basically is the average linkage that we are given. So we do it, carry out the clustering, then we have to plot the dendrogram, visualize the result. So basically, to the plot function, we give the result of the clustering. Labels, we want to label all the leaves with the proper labels. So the labels will be g dot lab, which are the labels that we extracted before. Okay. X lab, sub Y lab, we don't want any labels anywhere. Main is basically average linkage. We just want the title to be average linkage. And hang equal to -1, basically it keeps every all of these things at the same level, horizontal level. So all these names at the same horizontal level. So you can see when you are using average linkage, our tree is tree looks like this. And we can see that all the cells that are probably that are leukemia cells or melanoma cells or renal cells are like clustering together. And there's not a lot of intermixing between different cells. Okay. So next we will carry it out again with complete linkage. So average linkage is basically for each of the clusters, we are taking the average of the pairwise distances as the distance, cluster distance. For complete, we take the maximum of the pairwise distances. So again, we'll do that. So in this case, we are basically plotting the result. And inside this, instead of creating a variable and carrying out the clustering and then plotting it, we are doing it inside the plot function. So basically, whatever the result of this hclust will be there, will be provided to the plot function. So again, we provide the distance matrix. We tell the method is complete, and rest of the things are same. We just change the name to complete linkage. Now we carry this out, and we see that okay, our tree looks much more balanced. Like the center is somewhere here and all. So in the previous case, the center was slightly towards the left, and we see that okay, there's good separation between different kinds of cells, different kinds of cancer cell types. So this also probably is a good job. Complete linkage does a good job. Now we want to try it out again with single linkage. So basically we copy this whole line and we just write the method as single. We run this and we see that our dendrogram is slightly skewed. So basically it keeps on adding clusters, 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 which is also known as chaining. So it keeps on adding multiple clusters to the same cluster because there might be slight variance to that or something. So like the closer points are closer even though the clusters are far apart. Okay. And in this case, if you see that, okay, so for some of the cancer types, it has not been able to like uh, cluster them together. For example, ovarian cancer is spread around everywhere. Then there's a lot of intermixing in here, colon cancer, ovarian cancer, melanoma, and everything. Okay. So generally in biology, we tend to avoid using single linkage. Uh, Complete or average linkage is what we generally tend to use in biology. Okay. Does anyone have any questions regarding this? Okay, great. So we'll continue to our third algorithm, which is for classifying. So we learned about two algorithms for clustering. We also learned about two algorithms for classifying. 1 second. Yeah. So decision tree is our first algorithm for classifying basically. So in decision tree, what we have is basically, let's say you have data points like this. You have like a tree kind of structure here. Okay. Which takes decisions at each point. The first node here is called the root node or something. So each of these things is called a node. The first one is called the root node where everything starts. Then the final one, which basically give you a classification for the class or a label for the class or predict a label for the class are basically called leaf nodes. Okay. And the nodes that are in between these root and leaf nodes or root and internal nodes are basically called internal nodes. Okay. So that's it. So how does this work basically? So what this does is it basically divides the whole space into different squares and then not different squares into different smaller subspaces and then assigns a class to each subspace. So basically what we have here is G2 is less than equal to 3. We have a logical statement here. So what we do, we draw a line at G2 equal to 3 and we draw this line. So if the value of our point is less than or equal to 3, okay, if that is true, then we come to here. So for any point that is on this side of the line, which is less than equal to 3, 3 will go here. For any point that is greater than equal to 3, the answer here will be no and we'll go here. And the algorithm has decided that almost all the points in this half, this part are basically points of class 2. So it has assigned this region as class 2. So this half of the data is class 2. Then, so that's done. Then when we move to this side, we move to this node, this internal node, which tells us, which asks another question. G1 is less than equal to 4. So we draw another line here Notice that now we are just working with this half of the data. So each time we basically are splitting the data into two parts, and then in each part we are focusing on one part. In each half we are focusing on one part. Right? We are making two halves, not exactly equal halves, but two halves of the data in this case. Okay. This is basically a binary decision tree. So now we have this equation. So G1 is less than equal to 4. So if G1 is less than equal to 4, if the answer is yes, the model says that the prediction should be class 1. So any point that falls here will be labeled as class 1. And if the answer is no, then any point that falls here will be basically labeled as class 2. So now what we have done is we have basically divided our space into three uh, different subspaces, and in each of the subspace we have a class label that we want that we'll assign to uh, new data points, right? So okay, so one thing is in each of these steps, in at each of these nodes, we are asking some question, right? So here we are asking whether G2 is less than equal to 3. The value of G2 is less than equal to 3. Here we are asking whether the value of G1 is less than equal to 4. Right. So how do we know what question to ask? Okay. And even before that, so here this question is asked, G2 is less than equal to 3. There are two answers, yes or no. If you go to yes, there's another question. However, if you go to no, there's a label. So how does the model decide when to put a label also? Right. So these are the two things that we need to figure. So that needs to happen for a decision tree to work, to build a decision tree basically. So basically what we calculate is the main goal of the decision tree is, so here we have G2 is less than G3 at this point. We have all the points in our data set. Okay. We have not divided it into any subspaces. So we have all the points. We have all the points for class 1, all the points for class 2. It's more or less homogeneous, right? Sorry, it's more or less heterogeneous because there are many points for class 2, many points from class 1. Okay. When we divide it into two, and G1 is less than equal to, so G2 is less than equal to 3. Based on this, we are dividing into first this half and then this half. Okay. So this half has a lot of points from class 2 and very few points from class 1. So basically, it's a very homogeneous subspace. That's why it is assigned a class here. However, this half here is still not that homogeneous. There's still a lot of points of class 1 and class 2 mixed. So we need to divide it further so that it is more homogeneous and then we can assign a label. So when it goes beyond some certain threshold of being homogeneous or something, the model assigns some label to it. Okay. So how do we measure this homogeneous or heterogeneous thing in the data? Okay. So we have different measures of purity for that, basically. Right. So two of these are discussed in class. So one is called Gini index and the other one is called entropy. So Gini index for a given region. Okay. Uh, so these are like different regions in our data. So when we draw this line, we divide it into region 1, region 2, then we divide it into region 2, region 3, and so on. So in one region, we have a Gini index for each region, so which is given as fmj into 1 minus fmj summation oj from 1 to k. So k is basically the number of classes that we have. So in this case, we just have two classes, class 1 and class 2. So k here will be 2. Fmj is basically what is the fraction of observation of class j in the mth region. So let's say we are talking about this region, this upper region here, this part, right? So what is the fraction of, yeah, f? So this is, let's say, region 1. This is region 2, this is region 3. Okay. if region 1 class 1, okay, will basically be equal to what is the number of class 1 uh, data points here divided by number of class 1 data points plus number of class 2 data points. So divided by total number of data points. So basically fraction of observations that are present here. Similarly, Fi2 will be equal to number of observations of class 2 divided by total number of observations. Okay. And then what we are doing, we are multiplying fmj with 1 minus fmj and then summing it over across all the classes. Right. So now let's say we have one region in space. We have two classes. Okay. We are doing some clustering. We have class 1 and we have class 2. And in one region, let's say we have 0 observations of class 1 and all the observations of class 2. Right. So the fraction of observations here is 0, fraction of observations here is 1. And here we have fraction of observations as 1, here we have fraction of observations as 0. This is one region, this is second region, this is third region, and so on. So in another region we have 0 0.5, 0 0.5. Let's say we take these three examples. So in this case, what will be the Gini entropy? Gini index. So the Gini index in this case will basically be, what we do is 0 into 1 minus 0 plus 1 into 1 minus 1. Because this is 0, we put it in this equation. This is 1, we put it in this equation. So this is basically Gini of region 1. So this is region 1, 2, 3, let's say. Gini of 1 is this. So this is 0, this is 0. So the Gini index here is 0. Similarly, for this, it will again be 0. If you want to find out the Gini index for this, G3, G2 will again be equal to 0. G3 will basically be 0 0.5 into 1 minus 0 0.5 plus 0 0.5 into 1 minus 0 0.5 is basically equal to 0.5 into 0.5, 0 0.25 plus 0 0.25 equal to 0 0.5. So this value here is 0 0.5. Now, if you take all these values and you take different proportion of points and you take these values and you plot it, you get basically something like this. Okay. This is basically the Gini index, and this is basically proportion of points in points of class 1, let's say. Okay. Now, as you increase the frequency of class 1, so when the frequency of class 1 is 0, which means all the cases are for class 2. Okay, now this is a binary classification. So all the points are from class 2, then you get Gini index 0. If all the points are from class 1, it's very homogeneous, you get Gini index of 0. Sorry. However, if there is intermixing, then you get a Gini index value which is greater than 0, and you get the maximum value when the proportion is almost half in case of binary classification. Hmm. Okay. Similarly, we have another measure of purity, which is called entropy, which also has a similar formula. So here we take log of the fraction and multiply with the fraction and then sum it off. Instead of the Gini index, which is much simpler, we just subtract 1 from it. Here we take the log. Sorry. And if you plot it and see the entropy or the H of different H value also uh, looks very similar. It also has a parabolic curve like this. And if you have very homogeneous mixtures, which is like 0% proportion of C1 or around 1% proportion of C1, then the entropy value is very low. However, if you have a very heterogeneous population, the entropy value is high. Okay. So what happens is when you reach a certain value where your entropy or your Gini index is very low, you know that the class is very homogeneous there. So for example, let's say the Gini index here is 0.47, the Gini index here is 0.45, and the Gini index here is basically in this region, it's basically, let's say 0.07 or something. Okay. So in this case, uh, the model has decided that 0.07 is a very low value for Gini index, so we can stop here. So we'll just assign a label here. Okay. Here, however, the value here is 0.45, which is very high. So the model has decided to go further. Okay. So you can use the Gini index or the entropy. So that answers just part of the question, which is when to stop. The other part of the question we had was what questions to ask here. So since here G2 is less than equal to 3, why not G2 is less than equal to 4 or G2 is greater than equal to 3 or something like that, greater than equal to 2, right? So for that, what we need to do, we need to ask many questions and then compare them based on something. So what we compare it based on is basically information gain. Okay. So let's say you had, I'll write here. So let's say we have G2 is less than equal to 3 and we divide it into two halves, right? Then let's say we have an alternative question, which is G2 is less than equal to 4, right? And then based on that, we divide it into two based on yes or no, yes or no, right? So now what we have, we'll, in this case, we'll calculate all the gene index or the entropy. So let's say we are calculating the entropy here. The entropy here is basically, let's say 0.47. The entropy here is 0.45. The entropy here is 0.07. And we do it here, h is equal to 0.47, h is equal to 0.45, h is equal to 0.40 or something. Okay. So what we need to do now is we need to calculate something called information gain, which is basically what is the entropy of the parent minus some fraction into what is the entropy of child 1 plus minus some fraction into what is the entropy of child 2. Okay. So what is this fraction? This fraction is basically, this is the number of proportion of points in C1, class 1. So basically this child 1. This is basically proportion of points in 1 second. Yeah, this is basically proportion of points in child 2. So you do this, you calculate this, the information gain here, and you calculate the information gain here. And let's say you find out that the information gain here is higher. So you choose this question. Okay. So when you're choosing a question, you tend to maximize the information gain from that question. Okay. So this is basically how a decision tree is formed. So what we do is initially, let's say we see all of the data, we want to ask what we want to figure out what question to ask. We'll basically ask random questions, then calculate the information gain and figure out which is the question which has the most information gain, then ask that question. Then we get two child nodes. Then from those two nodes, we'll decide if the Gini index or the entropy is low. so that we can stop. Otherwise, we'll continue. We'll again compare between different questions. We'll figure out a question. We'll ask it and so on. So we get a decision tree, basically. Okay. So how do we use a decision tree now? So once we build a decision tree, so let's say we want to figure out this new red point, which is basically G1 is 3 and G2 is 2.5. Where does it fall? So what we'll do, we'll see our tree. We'll start from here. We'll ask the questions that come. So the first question is G2 is less than equal to 3. So G2 is 2.5, which is less than equal to 3. So we select yes and we move here. Now we are asking G1 is less than equal to 4. So G1 is 3, which is less than equal to 4. So again, the answer is yes. So we come here. And now we don't have any questions to ask. We just have a label. So our point here should basically belong to class 1. So if we again draw the boundaries that we had drawn, so this was one boundary, this was one boundary, sorry. Uh, this is basically the other boundary. So this is basically our class 1, this is our class 2, this is our class 2. So basically we can visually see that our point falls here, so it's classified as class 1. Okay. So the issue with using a decision tree for classifying is that generally a decision tree has very low bias. So it can fit very well to a training data. So let's say we had some more intermixing, then our decision tree would be more longer. So let's say we had like, uh, for example, data like this. So let's say this is class 1, this is class 2, this is class 1, this is class 2, class 1 and class 2, and class 1 and class 2. So a decision tree, what it would have possibly done is divided it like this, then this would have been one class, and divided it like this, like this, like this. So basically now we have more number of decisions to make. So a tree will be more longer. Basically, these number of branches and all will keep increasing and we'll arrive at a solution. But our tree is very well fitted to our training data since it has all this low bias. So it can basically draw arbitrary boundaries between 2 points by going deep enough. So basically, it fits very well to training data. So it suffers the problem of overfitting. So it will basically have less predictive capability when we provide a test data set to it, which it has not seen before. So the solution to get rid of this is to balance between the bias and the variance of the model. We can do that for decision trees using two techniques. One is boosting, and the other thing is called bootstrap aggregation or bagging, which is a much common approach. So we'll look at one example of bootstrap aggregation, which is random forest classifier. So basically, a random forest classifier does two things. It's a decision tree model, which basically does two things. The first thing is bootstrapping, and the second thing is aggregation. So bootstrapping is basically, so for example, let's say on the left-hand side here I have drawn, you have a data set of n into p. So n number of data points, capital N, and capital P number of predictors or features for your data, right? So in bootstrapping, what you do, you take various subsections of this data matrix. So you get S1 into M1, where S1 is like a subset, basically S number of samples instead of N number of samples. And M1 is basically M1 number of samples instead of P predictors. So we take some of the samples and some of the predictors. And you do so by sampling with replacement. So you would take something, you replace it back, then you sample again. Okay. So we repeat that process. So we do it once, twice, thrice, and how many ever times we want. So each time we'll get a different data matrix, which is basically a subset of our data matrix. And then what we'll do to each of the data matrix parts that we get, we fit one model. So this will be a decision tree classifier 1 will fit to this. Decision tree classifier 2 will fit to this. Decision tree classifier 3 will fit to this. For example, if you want to do 3 cases here. So this part of the process is called bootstrapping. So we are basically making more sampling from our whole data, and we are making different parts or more replicates from it. Okay, random replicates. Then aggregation is what we use to predict the class of a test sample. So let's say now you have a test sample. You run the test sample through all of these models, DC1, DC2, DC3, all of them. And then you get some prediction. So let's say the first model predicts is from class 1, second model predicts is from class 1, third model predicts is from class 0, other models predict from class 1, 0, so on. So you get multiple predictions for the same test data. Then what you do, you basically take what is the majority vote from this. So let's say you had 100 of this decision tree classifiers. 95 of them predicted that it's from class 1, 5 of them predicted that it's from class 0. So you predict that the data point comes from class 1 because majority of your models are saying that it's coming from class 1. Okay. So a random tree classifier basically builds a random forest classifier basically builds a forest of different decision trees, and then based on a majority vote from them, it decides where your point should be classified into. Right. Does anyone have any doubts? Okay. So next we look at another type of classifier, which is called a maximal margin classifier. So basically, let's say you have points around here. So this is our class 1, the circle, and this is our class 2, the square, the rectangles, right? So what a maximal margin classifier will do, because it's a 2D space, it will find a line that divides our sample into two different classes. Okay, so this line is basically called the boundary, decision boundary. Huh. And then we have these dotted lines, which is basically what are the closest data points to the boundary. So on this side, we have this as the closest data point. On this side, we have these two as the closest data points. Sorry. So let's say all three of them are at the same distance from the line, which is basically M. And M is called the margin. Okay. So what does the maximal margin classifier do? It tries to, so M is basically the distance between the closest data point and the boundary, right? So the maximum margin classifier basically tries to maximize this margin, maximize the distance between the boundary line and the nearest data point. Okay. So in general, when you do it in 1D or 2D, uh, a maximal margin classifier tends to, like the optimum position of the decision boundary tends to fall almost halfway between the closest positive and negative, or like closest points of the two different classes. For example, these points are the closest point in the two classes, and the boundary goes somewhere in the middle of them, maintaining the same distance from each of them. Okay. Now these points which fall on the marginal line, which are basically forming the margin, which are basically influencing the boundary line, are called support vectors in a maximum margin classifier. So support vectors are basically data points that are deciding the decision boundary. So the boundary that we are making is based on just these 3 points, none of the other data points. So for example, if you move this data point from here to, let's say, here, the boundary will not change because the boundary is not dependent on this. The boundary is only dependent on these 3 points. Okay. Other thing is they are always on the border of the margin, and yeah, other data points do not affect the decision boundary. Okay. So this is about maximal margin classifier. However, again, the limitation of this is that it tends to overfit to the data. And if there is slight change to the training data, it can lead to huge changes in the classifier. For example, let's say you if you move most of these data points, nothing will happen. However, if you move this data point slightly nearer, so let's say you move it here or something, then okay. So now this becomes the closest uh, point to the boundary line. So the boundary line will again tend to shift this way to go farther from this, right? So it will lead to changes in your boundary line. The other thing is, if there are samples near the boundary, for example, let's say you have this decision boundary, you have a margin, this is basically your class 2 point, basically your class 1 point, right? So this is our support vector. So let's say you have a point which is basically of this class, but it falls somewhere here. Okay, slightly over the boundary. So the maximal margin classifier classifies this as a circle. This is basically this class. However, this is not. So it tends to misclassify samples that fall within the margin or close to the boundary. Right. So how can we improve on that? To improve on maximal margin classifier, people came up with support vector classifier. So a support vector classifier basically uses the principle of maximum margin classifier, but it does not have a hard margin. It has a soft margin. Okay. What does it basically mean? So in the case of maximum margin classifier, the margin needs to explicitly differentiate between all the data points. So class 2 will always be on this side and class 1 will always be on this side of this boundary line in maximum margin classifier. But it can never be that there are intermixing of classes after you have trained your data, trained your model on your data set. Okay. So in case of support vector, they don't have such hard rules for the boundary. So what happens is in support vector classifier, it allows some amount of tolerance to things like points falling on different sides of the hyperplane. So for example, most of the points for the squares are here, but one of them is here, and still it's okay for the support vector classifier. Okay, it allows a few points to be inside the margin, and it also allows a few points to be on the other side of the wrong side of the plane. Okay. Yeah, so that's it. So support vector classifier has a parameter C, which can basically restrict how much of this freedom you allow. So how soft is the margin is something that you can regulate so that you can build a better classifier. So the advantage of this is that it does not overfit to the training data and it can handle nonlinear problems to a certain extent. But what if you had some very serious nonlinear data? So for example, let's say uh, you have some data like this, let's say. Okay. And then you have this. So basically the X's are one class and the circles are one class. So here the support vector classifier and the maximal margin classifier will tend to like make a single straight line, a plain line, a line. However, any line will not fit this data. The decision boundary for this data should look something like this, right? The boundary for the data would be like this so that it can separate the two classes. But we cannot get this nonlinear form of expression till by using a support vector classifier or a maximum margin classifier because they both build lines only. So what we do is we use a trick in support vector classifier, we name it as a support vector machine. So basically what it does, it basically transforms your data into some other uh, space. For example, you have 2D data now. So let's say you have X and Y here. What it will do, it will transform it into a 3D space. So let's say we have a equation Z equal to X square plus Y square. Okay. So now based on that formula, what we'll do, we'll plot our data again, but now in 3D. We have X, Y, and we have Z. So our data should look something like, sorry, we have X here, we have Y here. So our data was looking like this. So it will look something like all our X's will be here somewhere, and all our circles will form like a circle around. So basically, this is at a separate plane than this, right? So now we have created a separation where we can basically have a straight plane pass through the whole thing, which is the decision boundary for this. So in general, when you have a maximal margin classifier or a support vector classifier or support vector machine, the support vector classifier, the boundary is basically for one-dimensional data, the boundary is a point. For two-dimensional data, the boundary is a line. For three-dimensional data, the boundary is a plane. For n-dimensional data, the boundary is in general a n minus one dimensional hyperplane. Okay. So in case of three dimensions, we'll have a plane right here, which is basically cut through this whole three dimensions and divide our space into this one cluster and this other cluster. Okay. So this support vector machine, what it does, it basically uses mathematical functions which basically transform your data so that you can find a non-linear decision hyperplane. So after you increase the dimensionality of the data, then what you do, you fit a lower dimension line, then you transform it back. So after we fit a model like this, if you want to represent it in 2D again, so our data will again in 2D look like this, right? And it will have circles all around. And our decision boundary will now look like this in 2D. And we'll have a margin here, we'll have a margin here, right? So support vector machines have three kinds of kernel. Linear will basically make a linear transformation. Polynomial basically does polynomial transformations, and a radial basis function can basically do higher order transformations, which basically help us build arbitrary shapes of clusters, shapes of arbitrary decision boundaries. Okay. So that's it for what was taught in the class. Does anyone have any questions regarding this week's lectures? Okay. So we'll move on to the question. So the first question is, which of the following is correct for the k-means clustering algorithm? So we'll read through the option. The first option is the algorithm uses the pairwise distance between data points to define clusters. Okay. The second option is the algorithm uses the distance of each data point from the cluster center to define the cluster. The third option is the algorithm uses cluster-cluster distance to define the cluster. The fourth option is the algorithm uses a maximum distance between clusters to fuse two clusters and create a new one. So k-means clustering, what we basically do, we have these different points. We randomly assign cluster centers. Then we find the distance of each data point from the cluster center and then define the clusters again. So basically what we do is we are doing this step B. So basically when we are doing k-means algorithm, when we are using k-means clustering algorithm, what we are basically doing in some of the steps, we are calculating the distance of each data point from the cluster center, and then we are defining, okay, this point belongs to this cluster, these set of points belong to this cluster, and so on. Okay, the answer here is B. Okay, the next question is, we are using the following code to perform k-means clustering in R. Clust equal to k-means data 5 and start equal to 50. W is clust dollar total dot within SS. What is stored in W? So W is basically, so clust is basically the result of the clustering. We have provided the data and we want 5 clusters. W is basically clust dollar total dot within SS, which is basically the total within cluster sum of squares. So the answer here will be C. Total within cluster sum of squares. Okay. The next question is, we have a gene expression data set for 300 genes in 40 tumor samples. We perform data clustering to cluster the tumor samples based on their gene expression signature. A part of the code is being given below. Clust is hclust d method equal to average. Which of the following statements is correct? D is a 1 by 780 vector. D is the Euclidean distance between the genes. D is a 40 by 300 matrix. D is the original gene expression data for all the samples. So we know that when we are using the H-cluster method, D is basically the pairwise distance between all the genes. Okay. So if we go back again and we see, right, if we when we are running this, when we are using H-class, we are using it on G dot dist. G dot dist is basically pairwise distance between all the observations, right? So the options here are D is 1 by 780, D is Euclidean matrix, D is 4 by 40 by 300, original gene expression. So the answer is not D. It's not the original gene expression. Although D is the distance between genes, is the pairwise distance between the genes. Since we are not given the code that produces D, we don't know whether it's the Euclidean distance. So we can't be sure about this option. So if there's no other option, we'll come back to this. So then what we need to do, we need to figure out what is the shape of it, right? So we'll go back again and see what the shape was when we were doing it. So g.hc is just scale data. This is our scale data. So this is basically 6830 into 64 is the shape. G.dist is the distance, which has the shape of, which is basically a vector from 1 to 2016. So since we have 64 genes and we are looking at the pairwise distance between all these genes, sorry, since we have 64 samples and we are looking at pairwise distance between the samples, what we get is 64 into 63 by 2, n into n minus 1 by 2. That is the total number of such combinations we can have. So here we have in the question, we have 40 tumor samples, right? So what we'll get is the number of such pairwise interactions we'll get is 40 into 39 by 2. So n into n minus 1 by 2, right? Number of interactions, which is basically 2, let's cut 20, 39 into 2 is 6, 7, 80. So basically it is a 1 by 780 vector. So the answer is this, not this. Okay. Does anyone have any questions now? Okay, great. So the fourth question is, which of the following statements is correct for a decision tree classifier? So decision tree classifier is a method of unsupervised statistical learning. Okay, this statement is false because decision tree classifier is a classifier and it needs some data, label data to learn. So it's a supervised statistical learning method, not an unsupervised. A decision tree classifier is a tree that should be read from the leaves to the root. Is again false. If you look back at the decision tree, when we are reading a decision tree to make decisions, we are basically reading it from top to bottom. And the top part is basically the root. We are reading it from root to leaf, not leaf to root. Okay. So this is also false. The third is the number of internal nodes in a decision tree classifier is a parameter and is decided beforehand. So this is false. The number of internal nodes is not decided beforehand. Depending on how you are dividing your data and based on the values of entropy, information gain, and gene index, you decide how many internal nodes. So the algorithm itself arrives at some number of internal nodes. Okay. The fourth option is the question asked at an internal node of a decision tree classifier is decided based upon the information gain estimated for that question. So this is the correct answer. This is basically what we do when we are building a decision tree classifier. So in each step, we analyze the information gain and then decide on a question. After we have the question, we make, so basically it's a yes or no question. It's a binary question. We get two child clusters. For two of those child clusters, we calculate the Gini index and everything. And we see that if the Gini index is too low or if the entropy is too low, we can basically label it as a class and stop there. Right. So that is basically what a decision tree classifier does. So the fifth question is we use a decision tree classifier to classify data into two classes. At an internal node, we have assigned 40 samples of class 1 and 10 samples of class 2. Calculate the Gini index at this node. Okay, so we are calculating the Gini index, which is given by this. So first we need to find out FMJ, so which is the fraction of observations of class J in the mth regions. Sorry. So since we are just making a calculation as for a single node, we'll drop the M. We don't need to deal with M region, X region, whatever. So G is basically given as f of class 1 into 1 minus f of class 1 plus f of class 2 into 1 minus f of class 2. This is basically our Gini index. Now, what is f of class 1? F of class 1 is basically number of samples in class 1, number of samples in class 1 divided by number of samples in class 1 plus number of samples of class 2. Right. So it is basically 40 divided by 40 plus 10, which is 40 by 50, which is 0.8. Frequency of class 2 is basically fraction of class 2 is basically number of observations of class 2 into number of observations of class 1 plus number of observations of class 2. Is basically 10 divided by 40 plus 10. Is basically 10 by 50, which is 0.2. Right. So now what we'll do to calculate the Gini index, we'll just put these uh, values here. So it will be FC1 is 0.8 into 1 minus 0.8 plus 0.2 into 1 minus 0.2. So it basically boils down to 0.8 into 0.2 plus 0.2 into 0.8. So it's basically 0.16 plus 0.16, which is 0.32. Yeah. Does anyone have any question? Okay. So the answer here should be the Gini index is 0.32 for this particular node. Does anyone have any questions? Okay. So the next question is, which of the following distance measures is usually avoided in hierarchical clustering as it may produce a skewed dendrogram? So as we saw before, this is a very straightforward answer to it. It's actually single linkage. When you use a single linkage in your distance measure for hierarchical clustering, You basically get a skewed dendrogram, and there tends to be a lot of chaining where clusters get added to the same cluster. Okay, and that's something that people tend want to avoid because it produces a very skewed and unbalanced dendrogram. Right. So that's it. The next question, which is the last question. So we have four data points, D1, D2, D3, and D4. The following diagram is obtained by hierarchical clustering of this data using complete linkage. Okay. The following matrix shows the distance between four data points. Identify the yellow colored data. So the important word here is complete linkage. So we know how to find the distance between clusters, right? We have four data points, D1, D2, D3, D4. We are given the distances between them. So note that the diagonal is 0 because distance between D1 and D1 is 0, D2, D2 is 0, D3, D3 is 0, D4, D4 is 0, right? And note that it is symmetrical because distance between D1 and D2 is the same as distance between D2 and D1. So we'll just focus on the lower triangle for easy for our ease. So what we need to do here is initially we need to figure out which of these points has the lowest distance. Okay, in hierarchical clustering, we need to figure out the data points which have the lowest distance, then we need to combine them, right? So the first thing here is this is the lowest value that we get. So basically 1. The second lowest is 2. So 1 is between D2 and D4. So what we need to do is basically we should combine D2 and D4 to make a cluster. Okay. So since D1 is already provided here, we can't write D2 and D4 here, right? We know that the tree should look like this. So what will happen? Basically, either this is D2 or D4, and this is the other one, D4 or D2, right? So most likely these nodes are D2 and D4, and this node should then be D3 because that's the only thing that's left. Okay. So we are asked to identify the yellow color data. So we arrived that this is D3. We should stop here and the answer is B, D3. Okay. Okay. So that is it for this week's class. Does anyone have any question regarding any of the questions that we discussed or any of the material that was discussed today for week 7? Does anyone have any questions from previous live sessions also? Okay. So yeah, I'll be here for some time. If anyone has any questions, please uh, do ask regarding this week or any of the past week, uh, whatever you may have. Okay, so I'll tell this again. So some people might have missed. So we have one more session on 16th March, which is Saturday, where we'll discuss week 8 lectures, the final week lectures. Then we have another revision session on 20th March, which is Wednesday from 6 to 8 p.m. If anyone has any doubts, so I would prefer it to be more of a doubt clearing session. So if anyone has any doubts, then uh, that's a great time to ask right before the exam. Otherwise, also we will, if uh, people don't have a lot of doubts, we'll go through all the materials in brief again. Uh, so we'll just touch upon what all was taught in different weeks and what are the like main points from that. Right. Yeah. Thank you, guys. So, yeah, we'll stop here. No one has any more questions? Okay, thank you. So we'll close.